Hi, my name is Neil Desai, and I will be going over anticoagulant and antiplatelet therapy in this chapter of cardiovascular disease as part of the NAPLEX review course. Here are some of the topics that's covered as part of cardiovascular disease state goes. You could see some of the medications do have overlap between, different cardio disease states. So as of this lecture, we're going to go over the anticoagulation, antiplatelet therapy. So here are the differences between the anticoagulants and antiplatelets. You can see here there's a coagulation cascade, that's anticoagulants, where they play a role in um, preventing fibrin clot formation, and antiplatelets prevent platelet plug formation, so they will actually bind to the platelets, whereas the anticoagulants will bind to the proteins that are used to cause the clot in the blood. So you can see here some of the common uses here, like DVT, PE, AFib, stroke prevention, mechanical valves, DVT like deep vein thrombosis, peripheral embolism. You can also see as antiplatelets being used for myocardial infarction, unstable angina, post-stent, stroke prevention. So these are just some of the areas where these can be used. I listed two diagrams down here just for your reference, showing how the clots are being formed within the blood vessel. You can see here the anticoagulants being used, and a slow form, and then antiplatelets, a fast flow. Um, that's because, antiplatelets platelets generally just work straight in the bloodstream. So it's a little bit faster, so you could just go directly through. Whereas the slow flow looks like it's going through the proteins that will directly interact with these clotting factors and all of that. So it goes through a longer process. So that's a slow flow here. Um, so antiplatelets are generally faster, but sometimes both of them are needed together to successfully prevent that clot. There's some cases when both of these are needed. So we'll look at more of that more in detail later, but for now we'll just look at basic mechanisms. Just to note a couple of things here. Clotting factors are specific proteins produced by the liver and dissolved in the blood plasma. So it definitely goes kind of slow because it has to go through a chain reaction or coagulation cascade to form these sticky long threads of fiber and platelet plug that to reinforce that platelet plug and trap more blood cells to form the clot. Um, so that's what clotting factors are. The anticoagulants will work against that clot by directly binding to these enzymes that um, prevent the clotting factors from doing its work and forming these stable, strong clots. And then platelets are like cell fragments that just circulate in the blood, like red blood cells and white blood cells. So they're essentially just like things that just go through the blood vessel. There is no coagulation cascade or some process that forms like fiber threads or anything like that. So anticoagulants I will be talking about here are unfractionated heparin, low molecular weight heparin, direct factor 10A inhibitors, direct thrombin inhibitors. You're going to see all of these in the same diagram. Warfarin, I put it there separately so you can see how that works. That one has a little bit of separate process compared to the other 4 bullet points here. And so you can see them here, unfractionated heparin or low molecular weight heparin, how they are binding to the AT3 enzyme, which is an enzyme that will cause like the thrombin to form, or antithrombin. And so you're going to see how it actually how it interacts with the 10A and the 2A clotting factors so that it can prevent that clot formation. So these forms of medications bind there, but direct factor 10A inhibitor binds directly to the 10A inhibitor. It doesn't have to go through AT3 process. Same thing with fondaparinux. It just has to go through AT3, but it indirectly binds to 10A inhibitor. So basically what happens here is this one changes shape and then it interacts with the 10A. So it might be a little bit longer process versus the unfractured heparin, low molecular weight heparin, and a direct factor 10A inhibitor. So unfractured heparin, like I mentioned, they prevent factor 2A and factor 10A from doing its work and clotting the blood. The low molecular weight heparin primarily inhibits factor 10A more than it does to factor 2A. So it does some of the factor 2A, but not much. It directly targets 10A. And then you have thrombin inhibitors too. Thrombin inhibitors just bind to 2A. And then this is a diagram showing how the warfarin works. Warfarin primarily works in the liver. They inhibit this enzyme that converts the inactive form of vitamin K, which is in its oxidized form, and converts to active vitamin K, which is reduced form. So what happens is warfarin inhibits VCORC1 enzyme that converts from the inactive to active form vitamin K. So that's essentially what happens here. And vitamin K is very important because what happens is it's used as clotting factors for 2, 7, 9, and 10. And what happens here is, and it blocks that recycling, so there will be less of the active form of vitamin K produced to synthesize these clotting factors for doing its work in the blood. And also the anticoagulant protein C and S, that's what warfarin is designed for as well. So it kind of blocks those areas. The next section I want to talk to you about is the antiplatelet agents like COX inhibitors, the P2Y12 inhibitors, and glycoprotein 2B3A inhibitors, the phosphodiesterase PDE inhibitors. In the first diagram here, aspirin mechanism, the way it works here is that aspirin works on the COX enzyme of the platelet. So remember I talked about antiplatelets? These are the platelets that go in the blood. So we're going to imagine the white circles here as platelets, and you can see on there are some receptors. These are going to be where the COX-1 and COX-2 enzyme receptors are located, and then you can see the aspirin is bound to it, so it can prevent that that plug platelet formation. And so whenever aspirin binds to it, thromboxane TXA2 is decreased, which will decrease the platelet aggregation. And then P2Y12 inhibitors. This one is a little bit different. Instead of working in the blood vessel, it does go through the liver. And the medication in this case is clopidogrel. Prasugrel is another medication in this same class of medication. They are both prodrugs, and they have to go through the liver and get metabolized and activated by a CYP enzyme. In this case, it's CYP2C19, where clopidogrel get activated, and then it inhibits an enzyme P2Y12. ADP enzyme, so that the platelet activation is inhibited. So it prevents that activation of the platelet. And so there won't be any clots there because of this decreased activation. Glycoprotein 2B3A inhibitors, similar to how the aspirin work, but in this case, it's a glycoprotein inhibitor. It does decrease the fibrinogen cross-linking, so you're not going to see a lot of fibrinogen there, and it will prevent that clot formation there as well. And then a PDE inhibitor. This one is interesting as well. So with this one doesn't work on the surface, it works inside the cell. So you can see here, PDE is an enzyme that's going to break down the CAMP and the CGMP and convert them to the AMP and GMP. So PDE inhibitors is going to inhibit or block the PDE enzyme from breaking down the CAMP and the CAMP and the CGMP. And by doing that, we're going to increase the concentration of the CAMP and CGMP, so it will decrease the platelet aggregation.